So what is the problem that we're trying to solve with data visualization? Okay, so why are we doing this? If we look at the problems that we're trying to solve with science, they're mostly wicked problems. Wicked has nothing to do with the Wicked Witch of the West or something, but it's actually a term that's used since 1973. When two people described it in political sciences. But the wicked problem is basically a problem that is very difficult or impossible to solve. It's too big. Right? Examples are how to solve poverty, or how to solve climate change. Even though there's quite a few different characteristics, 10 characteristics described initially by Rittel and Weber, they can actually be boiled down to three main concepts. One is that there's not enough knowledge or resources to solve the problem. For example, to make looking at climate change, for example, we could only solve climate change if, bad example. Let's use another one. That's too difficult. Imagine there's a problem that you could only solve if you had unlimited funding. If you want to solve poverty in the world and you'd had unlimited funding, well, that would be one way of solving it, whatever that would be. Another problem is, another example is, what if you could only solve a problem if you had the DNA sequence of every individual that ever lived? That's impossible to get that, right? So that sort of problem would then be a wicked problem because you do not have the actual resources to do this. Another aspect of wicked problems is you have very complex interactions with feed-forward loops, feedback loops, nested hierarchies and stuff. So that if you look at the figure that we looked at before, the factors that influence that water, land, and food resources, a lot of things are linked to each other, right? If you look at biology as well, so the transcriptome is an effect of the genome, but it's not just a one-to-one mapping. It also depends what tissue you're looking at. It depends on the time of day. It depends on so many different things. What the transcriptome looks like. That has the effect of the metabolome, and so on and so on. It's very complex systems. A third issue is that human values and norms actually play a role as well. And we have to take those into account. And actually, that's where politics comes in as well. For example, if the problem that you're looking at, if the problem is something like security in the streets, right? or the sense of security in the streets, what you could do is you could put more police officers on the streets, right? That might increase the sense of security. At the same time, it might actually decrease it because people may think, actually, there's so many police on the streets, so something's wrong. You may, the police that's already there, you may arm them, right? So you can give them heavier guns or more guns, for example, instead of just pepper spray, let's say. Or you could actually take their guns away because criminals are less likely to shoot someone who's unarmed, right? So it really depends what you do, what your humans, what your values and norms are, what the solution to a certain problem could be. Okay. Some very specific characteristics is that the solution cannot be tested before it's realized. So it's not that you can set up tests and then let's try 5 options and we'll use the one that works best. No, it's one go. You cannot do tests beforehand. Another problem is that there's no stopping rule, so you can't say now we solved the problem. Now we solved the problem of climate change. That doesn't exist. So if you look at wicked problems, so that suffer from finitude, complexity, and normativity, we're looking at really very open questions, right? Which is a little bit like what I mentioned in the beginning on that competition of that island south of Greece. That's a very open question. In that problem, the question was, what's going on? That's an open thing. It's not like, did this happen or did this happen? No, it's an open question of what happened. What was going on? So we cannot solve these ones. So we have to actually break that down a little bit. So from wicked problems, we go into complex problems where at least the problem itself can be defined. But we still cannot solve those ones because we cannot define the solution. So actually what we're working on are tame or engineering problems. That's what we call them. So instead of looking at how do we solve climate change, we're looking at how do we improve the efficiency of a certain engine with 25%. That's something that you can test, right? So let me wait a second. Voila. So instead of having a very open question, we're working with a closed hypothesis, right, in statistics and data analytics. Well, data visualization works in this area. So data visualization starts with a question, an open question. It helps generate hypotheses that then can be analyzed further later on. Okay. Also, data visualization is about insights that then lead to hypothesis. It's not about proof. It's not about p-values. A p-value is not, well, a significant p-value is not the same as insight. So basically what we need to do is we need to move from the reductionist view of doing science to a more holistic view. So what is, what do I mean with reductionist and holistic? With the reductionist approach, it's been very successful over the centuries, but at some point you run into a wall, right? So with the reductionist approach, you really try to understand the whole of a system by understanding the parts of the system. So just like biology is basically chemistry, which is basically physics, right? Just like a population of individuals consists of individuals. Those individuals have organs, those organs have cells, those cells have organelles, those organelles have molecules, and so on. So if you really can understand the molecules, we can understand the population. That's the idea, which obviously doesn't work. So we need to look at everything at the same time, so integrate everything together. One of the issues is that we have can have emergent properties. So some properties of a system only arise at a certain level of complexity. For example, you would not, you could not say that a single brain cell is thinking about that brain cell, about that person's grandmother, right? A single brain cell doesn't do that. That's the whole brain. A single H2O molecule is not wet. It's the collection of H2O molecules that are wet. So we need to combine everything together. And for that, we need to combine things that humans are good at with things that machines are good at. So, for example, we call that Fitts list, Haba Maba. Humans are better at, machines are better at. So humans are good at recalling relevant pieces of information, which machines are not. Machines are really good at multitasking, which is something that humans cannot do. And so on. So we really need to combine those two together. At the same time, as we move from data to information to knowledge and to understanding, the role of the human becomes more important. For each of those, there's different types of questions you can ask. For example, for information, well, if you just have information, you can ask things like who, what, where, or when. If we are talking about knowledge, then we're talking about the how. And if we talk about understanding, we talk about the why. And with each of those levels, the role of the human becomes more important. If you have the chance, read the book Weapons of Math Destruction by Cathy O'Neill. She has a background in data science, but she wrote this book which is very accessible and basically gives examples of where machine learning methods and statistical methods had very bad consequences and unintended consequences. Even though they were made with the best intentions, they were unconscious assumptions that basically led to different issues. So definitely have a look at that book. So one of the things that we also see is that, things that they mention is sensitivity of parameter space for data analysis and so on. We'll come back to that later as well. But one example is reliance on statistics for data exploration. So I don't know if you know the Anscombe's quartet, but this is similar. This is Anscombe's dinosaur. Basically, these are all different data sets that have exactly the same mean for X and Y, the same standard deviation of X and Y, the same correlation between X and Y, the same number of elements, the same regression line, and so on. But still, I would argue that those data sets are not the same. Some are linear, some are point clouds, some are circles, a star and a dinosaur. But still, there's so many data analysis reports or findings where they say, yeah, well, we had a look at the data and we described the data, and this is the mean and this is the standard deviation of that data. That makes no sense whatsoever. Another example is this. So this is also actual data. So just looking at this video at the top. So even though the data points in the middle, they change a lot, this box plot doesn't change at all. So there's a live box plot, but it doesn't change at all, right? The same with these ones. The same with this video at the bottom. So this plot doesn't change at all, even though the data clouds change immensely, right? So always have a look at the data instead of looking at summary statistics or summary plots. By the way, don't use box plots. We will come back to that later. So why you never should use box plots. Other things that are difficult to integrate is thick data. So everyone talks about big data, but there's also thick data, which can be very small but very rich. For example, interviews. So how do you include information from an interview in a data analysis project? That's very difficult to do. And in the end, what is the solution that we try to, what's the approach that we use? The approach is visual analytics. That's what this course is about. In visual analytics, we basically combine automated data analysis with data visualization. And very importantly, there's a constant loop between the two. So you may, for example, have a very high dimensional data set. Let's say you're looking at gene expression data, 25,000 dimensions, which are impossible to visualize. But you first do a dimensionality reduction, a TSNE, PCA, UMAP, whatever. And using that transform data, you can then visualize that data. Having that visualization, you can think of actually what types of analysis are necessary. So you do those analysis, you visualize again, and it's that feedback, hermeneutic feedback loop that is really useful, that then leads to knowledge in the end.